like to welcome you all. Today I'm pleased to welcome you to the SmithGroup lecture at the Beckman Institute. This long-standing lecture was generously established by the SmithGroup, formerly Smith, Hincham, and Grills Associate of Detroit, the architectural firm that designed this beautiful Beckman Institute a number of years ago now. At the completion of the project, the firm was particularly proud of the Beckman Institute and presented us with a generous endowment to support the lecture series that you're here to attend today. The gift was given in honor of our founding director, Ted Brown, who's still with us and still contributing to the Beckman Institute. In the background, well, maybe not. Okay, I'll leave that one out. We have a different background. It's our pleasure to have Andy Visano and Raina Lee from the SmithGroup with us here today. Could you raise your hands right there? The group that built, and Andy was involved in building this wonderful building that's still in great shape. People come here all the time and ask me how old it is, and nobody believes it's 21 years old, which it is. To begin with, I'd like to introduce a long-term Beckman Institute faculty member. I don't mean old, but I guess maybe, he's a little older than I am. Steve Levinson, but he looks pretty good. Steve is a faculty, I can't always go by script. I'm always off script. Steve is a faculty member in the Human-Computer Intelligent Interaction Research theme and a professor in electrical and computer engineering. He received his PhD from the University of Rhode Island a number of years ago and worked at Bell Labs before coming here as head of the Linguistics and Research Department. Steve's research interests include speech synthesis, speech recognition, automatic language understanding, cognitive science, and artificial intelligence. Like me, he never decided what he wanted to do when he grows up, I think. So his lab recently acquired a rare humanoid, not quite human, maybe someday, robot called iCub. And Steve and his students are working on teaching iCub new information. It's a self-learning robot. Steve has many honors and outstanding achievements, including being named an AT&T Bell Laboratories Distinguished Member of Technical Staff, being named an IEEE Fellow and a Fellow of the Acoustical Society of America, and many others. Please join me in welcoming Steve.
Well, it's really nice to see this great turnout for the Smith lecture. As I told Jeff, we ordered up this beautiful Indian summer day just for the purpose. It's too bad we can't open the curtains and let the outside in, but then we wouldn't be able to see the screen. I want to say a few words of introduction to Jeff. He specifically asked me in my introductory remarks to confine my comments to his recent research, about which he's going to speak today and of which he is very proud, and for good reason. But I would be absolutely remiss if I didn't say something about all of the work which led up to that. It turns out, I didn't realize this, I had a completely different impression, but from our discussions yesterday, it turns out that Jeff has a long time interest in brains and intelligence. And that was always his goal to study, dating back at least to his undergraduate days at Cornell. Along the way, he did some other things, and you may know that Jeff is the founder of the company Palm and Handspring, and these were absolutely bleeding edge companies that offered us the very first PDAs. And these also came from a profound insight that Jeff had, which he told me about the other day, and that is that there was a confluence of developments. Just at the right time, the battery technology, the display technology, and the microprocessor technology were just barely able to support what he believed was the vision of the future, namely that we would all carry around our computers in our shirt pockets and purses. And so he went forward on that when many other people, incidentally myself included, thought that it was either not desirable or not feasible. And of course you know that Palm and Handspring were very successful. He then founded the Redwood Institute, in order to get back to his long-standing interest in brains and minds. And the Redwood Institute is now under the same name at Berkeley. And Jeff is conducting his research in his newest company, which is called Numenta. And although you might think it's a small startup, it really is a wonderful small research lab. And Jeff has developed a lot of software, and much of which he has made publicly available. I'm sure he'll tell us about that. And he has told me that in the very recent time frame, a lot of the ideas that were originally explored at Numenta have been greatly expanded, and that's the subject of his talk today. I just want to say, be especially on the lookout for his notion of the hierarchical organization of brain, and the importance of the sensory motor connection to come up with sparse distributed representations of world knowledge in the brain. I don't think I need to say any more. We're very pleased to have Jeff with us here today, and please welcome Jeff Hawkins.
Thanks, Steve. Thanks, Steve. It's a pleasure to be here. Thanks for all coming out today. I sound okay, I hope. Sounds all right back there. So thanks, Steve, for the introduction. Thank Art. I'd like to thank the Smith Group for sponsoring this lecture, the Beckman Institute, which is a cool place, my first time here, and the University of Illinois. So as Steve mentioned, I've been interested in the issue of biological intelligence for over 30 years. That's one of the primary things you guys list on your website for the Beckman Institute. It's the #1 thing, so we have something in common there. And as he mentioned, although I've been in and out of it part-time, full-time for many years, believe it, when I started POM, I had in my contract that I could work part-time on neuroscience. So I was a part-time entrepreneur. I've been doing it more than full-time for the last 9 years, 3 at the Redwood Neuroscience Institute, and now almost 6 at Numenta. As Steve also mentioned, we've made some real interesting progress in the last 12 months. We have these learning algorithms at the core of our work, and we figured out a new way of doing them, and it sort of snowballed. And I'm going to talk about that work today. This is really the first. This week is the first time that we've presented this in any kind of depth. I can't say today is the first time because I gave a couple talks 2 days ago at MIT. But this is the first week that we're talking about this. There's a lot to it, and I just want to point out right now, I'll walk you through the whole thing, but the details of what I'm going to present are actually, we posted some documentation, very complete documentation on our website on Wednesday morning, and you can look for that there. Okay, so with that, let's get going and talk about the brain. I think when you deal with, especially an interdisciplinary group, it's very important to tell people why you're doing what you're doing and how you go about it. So I'm going to tell you that right now. I work on a few premises. One premise is that the principles of brain function can be understood. This is not something that's going to take another 100 years or 50 years. It's all complete within our grasp. We have a tremendous amount of detail here. This is our time to do this, and we can do this. The second is we can build machines that work on those principles. That is, we have the sufficient technology today to make machines that work on the same principles of the brain. We're not being held back in that regard. We're being held back on ideas. Finally, my third premise here is that there are many interesting and important machine learning, robotics, or AI problems that can only be solved in this way. I mean, by studying how the brain works and then implementing machines that work on that way. Not all problems are like that. There are many problems that don't have to do this, but there's many that are that way. And so to me, there's a huge potential to do good in the world, to build machines that are really beneficial to humanity and are going to be a very, very large business. So those motivate me, and maybe they motivate some of you. Our focus isn't on any part of the brain or the whole brain. We focus mostly on the neocortex, a few other things like the thalamus, but mostly in the neocortex. And I'm not going to assume any particular neuroscience knowledge here. So if you're probably familiar, the neocortex is about 75% of the volume of your brain. It's the wrinkly thing on top. All high-level vision, hearing, language, somatosensory. It's all done in the neocortex. Anything you can tell me about in the world is pretty much stored in the neocortex. And fortunately, the neocortex is built on a very repetitive structure. And that gives us hope that we can, even though it does all these different things, that you can understand how it works. And that repetitive structure sort of suggests it has common algorithms. And this was first proposed 30 years ago by Vernon Mountcastle, a physiologist, who said, you know what, even though the neocortex does all these things, I think it's doing them all in the same way. It's using the same algorithms. And that's pretty much become a most common belief, and it makes our job a lot easier. Now, I'll just make a contrast. There's some people I know study the cerebellum. The cerebellum is another part of the brain that also has a highly repetitive structure. But it's a very simple repetitive structure. And so basically 1981 or something like that, three researchers independently looked at the structure of the cerebellum. It has three basic cell types, and they could intuit what it might do. The neocortex isn't like that. The neocortex is a very complex repetitive structure, and if you look at it, you can't just figure out what it does. So it's very, very difficult to study and very, very difficult to look at. It also has a hierarchical arrangement, so as we'll see in a moment, which adds some complexity to the whole thing. The process by which we work, and I'm going to give you a very detailed model in a moment, and you might say, well, how did this detailed model come about? Did we just pick up a bunch of ideas and throw them into a box and say, how does it work? No. In no particular order, this is how we go about it. At any point in time, we have a set of algorithmic needs. These are things like, okay, I need to deal with certain attributes of sparse distributed representations, or I need to learn sequences of those, or I need to be able to do online learning, and things like that. And then we go look at the neuroscience. And we say, what is the, particularly anatomy and physiology, say, what is the structure of the neural tissue that does this, and what does it tell us about the nature of the solution, how the information must be stored, and so on. And then from that, we build a biological model, a hypothesis. Says, here's how I think the neural structure is doing this. Then we take that and we build a computer model. Now, the difference between the computer model and the biological model is the computer model is implemented on a computer, but we can't do everything the biology does. We have to take some shortcuts or corners or change things to make it work. But we try to tell you very true to the biological model, although it's not a simulation. We're not doing a simulation of the cortex. Finally, we run this on data sets where we get empirical results. And we come back from the empirical results and say, hey, it worked like we thought it did or didn't work. We can feed it back into our algorithmic needs. And if you start with this process, you add complexity, biological complexity to your model over time. I'll tell you, 12 months ago, our models didn't even include neurons. And now you're going to see in a moment, here's some very sophisticated neurons in these models. OK, now I need to give a little bit of neuroscience background. One last point here. Our models, in the end, you'll see, are biologically empirically driven. They're not mathematically derived. It's not like we start from mathematical principles. We use math along the way, but they're really biologically empirically derived. OK, let's start with some very simple biology here. The neocortex, there's a picture of the neocortex. It's a sheet of cells about 100 square centimeters, 2 millimeters thick. This is my favorite model of the neocortex. It's a dinner napkin. It's basically about this big. It's about this thick. It has somewhat, no one really knows, maybe 30 billion cells, neurons in it, maybe 100 trillion connections between those cells. And that is basically you, right? You are this napkin in your head. It's all shrinkled up in everything you know about the world and everything you do, and my speech and your listening is all happening in this sheet of cells. It's really quite amazing. The neocortex is divided into regions. And so if you look at the different areas, there's areas involved with vision and hearing and language and motor control and so on, and there's dozens of those regions. And if you look at in detail in those regions, the detailed architecture, they're almost identical. There are differences, and people debate this, but the overwhelming similarity is just amazing. What differentiates the regions is not intrinsic function, it's by connectivity. So the basic theory is, a hypothesis, it's generally accepted in the neuroscience world, is that what makes a vision area vision is because it's connected to the eyes or to other vision areas and so on. And we know from many examples that the thing can be rewired. You can feed information in different ways. So we have this, it's really all about connectivity between regions and not what the regions themselves are doing. And then, of course, that implies that there's these common algorithms underlying what's going on here, which I've already talked about. If we then take one step further and we ask ourselves, how are the regions connected to one another? And they are. So if I have two regions here on my sheet A and B, and there's a big nerve bundle of nerve fibers going from A to B, and B projects back to A, you can map out the feed-forward connections, and you can map out what the structure of this is, and you find that it's in a hierarchy. And this is a very famous picture on the left from Fellman and Vinessen, which is showing the visual areas in the macaque monkey, and it shows how those little boxes are regions, the colored boxes are regions of the monkey's neocortex, and it shows how they're connected in a hierarchy. In this case, visual information is coming from the bottom, and it moves up the hierarchy and down the hierarchy. We know that as you go up the hierarchy in regions that are further away from sensory input, that there is a convergence. There's always a convergence. Information is always convergence on the cells, and the cells represent larger and larger parts of the input space. Initially, larger and larger parts of the visual auditory space, but then multimodal and so on. There's another thing that happens, which is this temporal slowness as you go up the hierarchy. So cells respond to very short and fast things occurring at the bottom, and they tend to be on longer periods of time as you go up. So it's a temporal coalescence that's happening there. Okay, we have a basic theory. It's so basic, it's almost trivial in some sense, about what's going on here. We call it hierarchical temporal memory. Essentially, it's a very descriptive term. It says it's a memory system, it's hierarchical, and it's based on time. And we'll get into that. We can show it here like a picture. Here's a 4-level hierarchy. These would be like 4 levels, 4 regions in the neocortex. And information flows up and down the hierarchy. The basic theory says the following. What are we doing? First thing it does, it's all about learning sequences. What's happening here is we're learning sequences, and before it can learn sequences, it has to be able to take this very massive input, convert it into some sort of spatial pattern, common spatial patterns, and then learn transitions or sequences of those. It's like, a very simple example is like learning melodies. You know, you come in, you say, oh, that's the interval, here's the song, I've learned the sequence, and that's what each region in the hierarchy is doing. That's what every region is doing. And the second thing it does is it then forms a stable representation of those sequences, of those patterns. This is really what inference is all about, is saying this series of patterns is really the same thing, and it passes that sort of stable representation up the hierarchy. It's like the name of the song. And so as you go up the hierarchy, you have converging time and coalescing time. If information flows back down the hierarchy, you unfold it in time, and that's what I'm doing right now when I'm speaking to you. I'm taking some high-level concepts, and I just unplay these sequences. It may look like I'm making it up, but believe me, this is all memorized in my brain right now, and it's unfolding and unfolding as it comes down, and it produces this very complex temporal pattern, which is my speech. How do we understand the hierarchy? Why is there a hierarchy going on here? This is the simplest explanation I can give for you. The simplest explanation is the hierarchy is two things. It's an efficiency mechanism. It basically makes an intractable problem tractable. It makes something where we take, it essentially says it reduces dramatically the amount of time I need to train, and it reduces the amount of training I have to do. It does so by sharing representations. So you can imagine at the bottom of the hierarchy, I have simple representations, and they get more and more complex as I go up. So if I wanted to learn a new speech, for example, if it's in English, I find it fairly easy to do because I do not need to learn new words. I do not need to learn new concepts. I don't need to learn new phonemes. We share that stuff in new ways at the top. If I would try to learn a speech, the equivalent speech in a language I just don't know, I couldn't do it. It would take forever. The second thing the hierarchy does is, and it works on the same mechanism, it provides a means for generalization. So that I do not need to learn every possible combination of thing I'm going to see in the world. I can see something novel composed of previously learned components and know how they behave. So a very simple example is I could see a new animal I've never seen before, but if it's got scales, I might imagine it's going to swim. I imagine it's going to move in certain ways. It's going to have certain attributes. And I can adopt all those. So it's a generalization method, too. I'm going to talk here. Why did we come up? Why do I say sequence memory is really important? That was the question I want to get to here. Why is sequence memory so important? Where did this idea come from? How can really sequence memory be at the core of what your brain is doing? Let me give you some examples. First of all, we're constantly making predictions about the world. We're constantly making predictions of what you're going to feel and see and hear. It's a multifaceted prediction. We know this is true. And the only way you can make predictions is you have some temporal knowledge about what typically follows what. What will I expect to feel when my hand comes down on this lectern? It's not going to feel like milk or jello. It's going to feel like wood. And if it didn't happen that way, I'd be very surprised. I have expectations of what I'm going to hear and see and feel. Second of all, we know that all parts of the neocortex are involved in motor behavior. And motor behavior is nothing else. If it's nothing else, it is a series of sequential patterns of activation. So we have to have memories of how to do this. I have memorized how to say things and how to make these commands, and they're time-based sequences. Most of the inference we do in the world is time-based inference, meaning I can only infer what's going on by receiving a stream of data. So language is like that, hearing is like that, touch is like that, most of vision is like that. although we can do spatial inference in vision. So if I'm going to do time-based inference, I have to have knowledge of what things follow what. I have to have memory of sequences of patterns. And finally, spatial inference, which is sort of the idea that I can flash an image in front of your eyes and you don't move your eyes, nothing moves, and you can tell me what it is. That's actually also based on time, in the sense that in order to learn what something looks like, you have to see how it moves through time. And after the fact, you can say, oh, I saw different dogs moving like this. I've seen that this image and this image and this image are all the same thing. I can now group them together and I can show you one of them, and that's how you know that all those different images are dogs. The point of the theory here is that basically time is at the core of everything here, learning how sequences of patterns change in the world and how to do that. We know that some attributes of the sequence memory that we need to do in a hierarchical temporal memory. One is it has to have high capacity. We know a tremendous amount of things in the world. You can navigate this building, your home, your room, your apartment. You use computers. You can do an amazing amount of things you have stored in your head. So very high capacity. It's all very context dependent. What you predict is going to be depending on where you are exactly at this moment in time, which may be different than other times. We know it has to be robust because brains are robust, and the inputs are robust. So we know that brains can suffer all kinds of trauma and keep working, cells die. But we also know that our inputs are very impoverished and can get occluded. And I talked about predictions a moment ago, but one of the things is we want to make sure that our memory, our sequence memory, has to be able to predict multiple things at once. It's not good enough to say, oh, I know exactly what word you're going to say at the end of this sentence. That usually isn't the case. Usually you don't really know, but there's multiple predictions that are occurring. We want our memory system to be like that. We know that as I said earlier, we have to have a mechanism for forming stable representations of our sequences of patterns. It's very tricky to do, actually. And we know the system needs to be online learning. It needs to be able to learn while it's doing inference. So these are some of the attributes of memory. And it meant that we spend a lot of time figuring out what are the neural structures that do this kind of stuff. And I'm going to tell you what we think is going on. But before I do that, I have to give you another neuroscience lesson. So let's start with some biology. This is a very simplistic drawing, caricature drawing of what's going on in the neocortex. Again, we're taking our sheet of cells. We're now going to slice it, and we're going to look at the 2 millimeter thick slice anywhere in the cortex, pretty much. And what you find is there's typically 6 layers of 5 layers of cells. One's a connectivity layer. The cells are very densely packed in there, something like 100,000 neurons per square millimeter. They're massively interconnected, but they're laid out in these different layers. The cells in a column that is perpendicular to the surface tend to have a very similar response property. That is, they tend to have, they spend the same feed-forward input. They all seem to represent the same thing. And we can see this, many neuroscientists, when they're recording from cells, don't even bother to figure out which layer they're in because often, in some experiments, it doesn't matter. It's just basically, you're going to get a very similar cell response. Contrary to that, the majority of connections between cells, of which there are a huge number, are horizontal. They're the cells within the layer or an adjacent layer, but the vast majority of connections are sideways, not vertically, until you think that the cells maybe horizontally would be responding the same, but no, those are different. Finally, we know that the feed-forward connections coming into this region of the cortex are few relative to the horizontal ones, but they tend to be stronger. They tend to have a much stronger effect, and they determine the columnar response properties of those cells. Finally, we can say the following. Although there's 5 layers of cells, there's basically 2 that are primary feed-forward layers. And that's layers 4 and layer 3. So your classic example is you have some input coming from a sensor, a sense, or from another region. It projects to the layer 4 cells. The layer 4 cells project to the layer 3 cells. And the layer 3 cells themselves are the output that projects to the next feed forward into the next region. The contrary to that, there's a lot of variations of complexity in this, but one thing that's interesting is that layer 4 sort of disappears as you ascend the hierarchy. As you get further away from sensory organs, layer 4 goes away, and you're basically left with layer 3 cells as your primary feed-forward layer of connectivity. And we want to understand how these things are doing it. Now, we have an hypothesis. This is an hypothesis about that's underlying the mechanism I'm going to show you here. One of the part of the hypothesis is that these cells in these different layers are actually fundamentally doing something very similar. They're all going to be doing a similar sort of learning algorithm, and the cells are very similar. The pyramidal cells and the spiny stellate cells have a lot of similar properties. So each layer is doing something similar. Each layer is, I'm going to argue, learning sequences, doing prediction, and forming stable representations. And they're going to be doing that with sparse distributed representation. So we're always going to be working with some number of cells active out of a very large population, a small number active out of a large population. We're going to further speculate that we can understand the difference between layer 4 and layer 3 and why there are two different layers here and why if layer 4 goes away. And this will become clear in the talk, but basically layer 4 is learning first order transitions, meaning it's not using context. It's basically saying, given my input, I'm going to make a prediction what's going to happen next. This turns out it's ideal for spatial inference, meaning recognizing image shifted and rotated and scaled in different types of ways. It results in what people know as simple cells in say, V1. Layer 4 is also learning sequences, but it's doing as a variable order transition memory, meaning it's using context. And so it says, I know I'll make predictions based on context in the past. And that's ideal for time-based inference, and it leads to things like complex cells. And so you can actually start maybe piecing together, why does layer 4 go away? Because when you get away from sensory organs, you lose the need to do more spatial invariance, and you tend more to focus on temporal problems. Okay, you may say, well, what are all those other layers doing there? That's 2 out of the 5. I'm not going to talk about them, but layer 5 is primarily, it does a couple things, but one of it is the primary motor output for the cortex. So every region in the cortex has some motor output. They project subcortically to someplace else. And there's layer 5 cells that do that. That's also sequence memory. It has to do with specific timing. And 2 and 6 are hypotheses. These are related to feedback and attention, and I'm not going to talk about them more today. What we want to do is focus in on a layer of cells, getting at the core algorithm, how learning might occur to do that. I have to go one more level deep in the neuroscience here before we start talking about mechanisms. We need to talk about what neurons are and what they look like. Here's a picture of a neuron. This is classic. Many people would recognize this from Cajal. It's over 100 years ago. It's a classic pyramidal cell. And we can say some things about it. First of all, we've learned over the recent years or in the last decade or two that the dendrites, the branchy structures on it where all the connections are made between cells, is actually a quite sophisticated processing system. And we can break it into 2 groups. We can break it into the proximal dendrite segments, which I've indicated in green here. And if you can't see, it's just basically near the cell body. These act sort of like a linear summation. That is, inputs that come on synapses here are almost linearly summed in the cell body to determine if the cell body is going to fire or be active or not. They tend to preferentially connect to feed-forward connections. So we talked about how the feed-forward connections are few, but they're strong. Well, they're strong because they attach to the proximal dendrites, and they drive that cell very, very strongly. We can talk about the distal dendrites. These are the ones that are further away from the cell body. And we now know that they act on a very different way. They're much skinnier, and they act as sort of an integration zone. Each one of these, you can take a section, some small limited area of the distal dendrite, and there's some synapses on them, and that acts as its own little processing unit. And there's dozens of these sort of processing units in these regions. It's a nonlinear integration. Basically means that if a certain number of inputs are coming in at one point in time on a distal dendrite segment, that segment can become active. It's like a threshold detector says, oh, I've seen 15 active synapses at the same time. It can generate a spike down the dendrite and cause the cell body to do something. Those are primarily connected to other cells in the region and other cells in the layer. Finally, we can talk about, we can talk about the synapses. These are the connections between cells. There are thousands of typically on of synapses on the distal dendrites. That's where most of the connections are. There's usually hundreds or 100 or several hundred on the proximal dendrites. We know that there's all kinds of ways that these synapses strengthen and weaken and form and unform. One thing we know now we didn't know 30 years ago, for example, is that synapses can form very rapidly. They can be, it's not just a matter of strengthening and weakening them, they can form and they can unform. And in fact, that looks like how most of the learning is occurring, and that's how our models work. The output of the cell is a spike, but it's not really a spike. It's really a rate of spikes. And there's a lot of debate about this, but generally people say, hey, there's a variable rate that's coming out of this cell. It's some sort of activation rate. They also typically have often a small bursting mode, which is a different physiological mode, which is a very short type of spikes. And they project, they always come out and they split. And I show that on this picture here that they split. They send a projection someplace else, and they send a projection within the layer to connect to lots of other cells. So that's a real biological neuron. This is not a neuron. This is something that is used in a lot of very early and simple neural network models. And we don't want to do this. It's useful for certain types of problems, but it's not the kind of useful if we want to understand what a neocortex looks like. So all the early work in neural networks were something along these lines. You have a cell body, you have some weighted synapses, you have some input, you have some nonlinear function, you have a scalar output. That's not going to cut it. This is what an HTM neuron looks like. Again, I said I'm going to tell you this because we didn't start here. We ended here, and maybe we're not ended yet. It's very close to the points I made about the biological neuron, so clearly I would cherry pick to show you what I wanted to talk about in the biological neuron. But it's pretty good. I bet you not many people who know neurons in the audience said, hey, that's a bad idea. And they said, that's pretty good description. HTM neuron parallels the model neuron pretty well. We have a proximal dendrite segment, here shown with the green dots on it. Those are the green synapses. We model it as a linear summation. We connect it to feed-forward inputs. We map it as the distal dendrites is just an array or a set of distal dendrite integration units. We don't model sort of the topology of the cell. There's dozens of regions. They act as threshold detectors. So if I have over a certain number of active synapses, then that segment becomes active and the cell becomes active. We end up after learning, we don't start this way, but we end up after learning with very similar kind of numbers you see in the brain. We end up with thousands of dozens of distal, that's wrong. It says thousands of distal dendrites. It's dozens of distal dendrites. Sorry, I've skipped ahead here. There's dozens of regions, yes, the coincidence detectors. Synapses, we have thousands of synapses on the distal dendrites. There's hundreds on the proximal dendrites. And interestingly, we model the synapses as a binary thing. It's either connected or not connected. We don't actually have any further weighting on it. Synapses are pretty stochastic devices, so you can't rely on any kind of fidelity to them. So we say they're binary, but we do have the concept of permanence, which is essentially how permanent is the connection. And it can be completely unconnected, it can be growing, form a connection, and then be very permanent and hard to get rid of. The output states of our cells are different than real neurons. We found a need to have two basic output states. One is a sort of active from feed forward, meaning it's responding to a feed forward input. We can think of that as a burst, or you can think of it as a very fast activity. The second one is a predictive state, which is saying the cell is saying, I'm expecting to be active, but it's not really being, it's not receiving the feed forward input yet. And we also project laterally and forward, and there's more T deals there I won't get into. Okay, so what do we do now? We are now going to model a layer of cells in like layer 3 or layer 4 in a region of the neocortex. I'm not showing the hierarchy. I'm just showing one region, one layer, and we're trying to get at the learning rules and how we're going to learn sequences here. So I show a picture here. This shows a subset of a region of cells. Our models typically have hundreds of thousands of cells in them now. I'm showing one where the cells are arranged in columns that are 4 deep. So each of the little squares or boxes in that picture is just the cell body of one of our neurons. I'm not showing any of the connections. I'm not showing any inhibition effects. I'm not showing synapses. I'm not showing dendrites. I'm just showing the cell bodies there. So a region in our model is just a set of neurons arranged in columns. We're going to say the following things. We're going to just force the fact that all the cells in a column basically end up receiving the same feed-forward activation. In biology, we think this is done primarily through certain types of inhibitory cells. We just force it. We say they're all going to share the same proximal dendrite. But then the next thing is we say, but the cells in a column, they represent the same feed-forward input, but they represent that input in different contexts. So the cells actually differentiate in the context in which the feed-forward input is occurring. We need to be able to represent input in context. What does a region do? The region takes a very large, massive input. You can think of it coming from the retina, or you can think of it coming from another region, or from some sensory array. A lot of inputs, a lot of data, things, all little bits turning on and off. First thing it does is it creates a sparse distributed representation of the input. The input may not be sparse. It may be 50% of the bits are on, 50% off. We want to create a sparse representation. That's going to be key to how the whole thing works. Then we are going to create that, we're going to turn that representation into one in context. So we're going to form a new representation of it. Essentially, the feed-forward input is just the columns. Then we're going to pick a subset of the cells in those columns to represent that input in the context of prior inputs. And then we're going to learn sequences of those. We're going to learn sequences of the transitions between these sparse distributed representations. Once we've learned those sequences, we will then be able to make predictions. We'll then be able to follow sequences in time. And we basically have learned our sequence memory. So we'll be able to form a prediction on the current input in the context in a sparse distributed way. This prediction actually will typically be slower changing than the input. And the reason is that the prediction is multiple time steps in advance. It's like you're predicting multiple notes in a melody in advance. And that prediction changes slower than the actual inputs that are changing, and that becomes the output of the region. Okay, I'm going to walk you through some pictures to show you how this works. These images were actually from, this was actually from vision experiments. This is real data that we just visualized. It's not made up. In some sense, it's made up because we built an experiment, right? But this is from an artificial vision data set we're working with, and this is a subset of the cells. Now, I'm going to start with just showing you I'm showing you a layer of cells. Notice there's no column effect here. Well, I'm going to say this is one cell per column. I'm going to use the same rules, but I only have one cell per column, and then we'll switch to multiple cells per column. This will make it easier to look at. The different colorings of the cells represent the internal potential of the cells due to feed-forward input. So imagine you're a cell. You have some connection to the subset of input, which you're going to learn. I'll talk about that in a minute. And some input comes along, and everybody gets a different type of activation. Some will be more strongly activated by input. Some will be weaker activated by the input. And so the coloring here implies that. Now, what happens in real cells, the ones who get the most input are the ones who fire first. And what they're going to do is they're going to inhibit their neighbors. So what we're going to do here is we're just going to say, pick one of these guys, the top activated people, top activated cells. They essentially define an inhibition radius around them, saying, OK, I'm going to inhibit my neighbors who aren't as strong as I am. I got the most input. You got some less. I'm going to shut you down. And if we do this properly, that is, you say, there's some tricks to this because this area of inhibition has to change, and we have to set a percentage of cells. But what you'll end up with is you'll end up with a sparse distributed representation. This is a representation of an input space which is very, very large, and this representation space is much smaller, but it does a very good job at that. So now we have a set of cells. This looks maybe like 1% of the cells are active at this point in time, based on some complex input that went into this region. These cells are now the next time step. Imagine the next time step that comes along. I'll have a different activation. So that's time step 2, and this is time step 1. And what we want to do is when a cell becomes active, so imagine one of the cells here in the beginning, in the middle of this picture, when it becomes active, it wants to learn how to predict that it was going to become active. And so the best way to predicting to do that is to look at the activity that just occurred half a second ago, or a millisecond ago, or what was just going on. And so it's going to form connections to cells that are nearby that were just active. And every cell is going to do this. Every cell, whenever it becomes active in any representation, is going to try to form connections to things that were just active before it. And those connections don't form immediately. It has to see them multiple times, but if it sees them enough times, it'll form connections. And what will happen is you'll get an input like this. Every cell in the system now looks at this pattern and says, oh, is this something that predicts me? And those cells enter a predictive state. And so you end up with some cells that, the yellow cells here are all cells that are predicting what might occur next. This is a multifaceted prediction. You can see there's more yellow cells than red cells here. And this is basically saying, I think I know what's, given the current input, this is what's going to happen next. Now, with one cell per column, I don't have any context. Therefore, this is a first-order memory. Essentially says it doesn't matter what occurred in the past. Given this input, this is my prediction. And that's what it's going to do. So we're now going to switch to multiple cells per column. So the exact same problem here is that now we have 4 cells per column. This is now artificial data because it was hard for people to see what was going on with the real data because you can't see through all these cells. So I made it a little bit more dense here, but you'll get the same idea. Here's what it might look like if I have an input to this region, but there is no prior context. There was no expectation. And in this case, what we do is we say, hey, none of these cells were expecting to be active. And so when a column becomes active, all the cells in the column become active. So this is an input that's represented. And every single one of these cells you see here, there's this column underneath it that are all active. And you can see 3 or 4 of them on the edge here. The contrary position is if I completely expected this input, I knew what was going to happen, and it wasn't surprising at all, the exact same input would look like this. I'm going to show you the next picture is the exact same columns. So you can look at these 4 down here. But now it's only 1 cell per column. This is essentially saying, oh, I knew what was going to happen here. Well, it'll become obvious, but hopefully in a minute, how this all works. This is, I have a sparser representation now. Now think about it a little bit. Let's say I had 100 columns active at once here. and there's 4 cells per column. So very simple math says, how many different ways can I represent the same input? Well, it's 4 to the 100th power, which is a huge number. Right? It's one of those monster numbers, and when you deal with sparse distributed representations, you're always dealing with these very large numbers. So there is a huge number of ways of representing the exact same input in different contexts. And each one of those different contexts can lead to a different set of predictions or not. And so we have a tremendous capacity for encoding context here. And if you do the same process and you go through this, you'll end up with a prediction that looks like, well, okay, this is just saying it's, we have context and prior state. We end up with a similar sort of situation. Any point in time, you have the current input in the context of some variable distance time passed. It could be not just a previous step, but it could be any time in the past, you'll see. Then I can predict what might happen next. What are the possible things that might happen next? Now, let's see what happens when I have a new input in the context of a prediction like this. Look at, we're going to look at 3 different columns here. The left one here, the one on the left, is pointing to a column where the bottom cell was in a predictive state. The second arrow is pointing to a column where the second from bottom cell was in a predictive state. And it may not be obvious, but the third arrow here is pointing to a column that wasn't in a predictive state, that none of the cells are in a predicted state. So when a new input comes in, this is my prediction, the state of my system, and the new input comes in, what do I do about this? Well, let's say the new input invoked these 3 columns, among others. And so you see what happens here. In the new input arrives, it selects the column and says, oh, I was expecting this cell, and that's the only cell that becomes active. The second guy, same thing. I was expecting this cell. He already wins. The third column here was no expectation, and therefore it's a surprise. This was not an expected input. And so we can say, oh, all 4 cells are going to become active here. It's like they all burst at the same time. Oh, wait a second, this was unexpected. This basic idea allows you to, and I won't walk you through it because of the time limitations here, but allows you to do a very lot of sophisticated things in terms of temporal learning. It allows you to learn new patterns all the time. It allows you to detect when there's an anomaly. It allows you to detect how much of an anomaly is, where in the input space the anomaly occurred. And it leads to be able to morph between new statistics in the sequences of things change over time and the occurrences and so on. But that's the basic idea. Let's go back and look at our neuron and just walk through it again, exactly what's going on and some of the attributes that are occurring here. Let's look at the proximal dendrite, which is the green part here, right? And this is getting the feed-forward input. So as I said earlier, it's shared by all the cells in the column. So we're basically forcing all those cells to have the same sort of feed-forward function. As I mentioned earlier, it's a linear summation of the inputs. So they all just sum and they get some activation function. Now we had a problem. We found out there were some cells that never learned. They never won. There's a competition, and some guys just by chance didn't have the right stuff. And so what we did is we introduced the concept of boosting, which is essentially saying if a cell does not become active as often enough relative to its neighbors, it boosts it gets bored. We call it boredom. And it says, I'm going to be more aggressive. And it basically boosts its input until the point where it starts winning. And when it starts winning, it'll start learning something. And when it starts learning something, it starts pushing the other guys nearby out of the way. It says, I'm going to win, you're going to lose. And it will take over some part of the representation. This is really cool, and you'll see in a second why. The second thing we do is we form synapses from a potential pool. This is a concept that comes from biology. And the idea is that you have an axon and a dendrite. They may not form a synapse, but they have the ability to form a synapse because they're nearby. We've extended that concept to say that we want to be able to allow sort of dendrites and axons to grow. So we actually give every dendritic segment a very large potential pool. We give it a Gaussian distribution on this potential pool, so it's more likely to start in one's place. But what happens here is it allows cells to move and represent things differently over time. It allows us to do things, the plasticity you see in the brain. So what I call it here is self-adjusting representations. If I knock out part of the sensory array, all the cells will readjust to represent better what is coming in. If I lose a bunch of cells and they die, then the other cells will move in and represent what those cells did. It's very cool. It's just like what you kind of see, the plasticity you see in the brain. So that's an important part of this, is that the input is actually the representations that are formed are sort of self-adjusting. So they're dynamically determined by both the resources you have and the statistics of the data, and they can morph into new representations over time. The distal dendrite segments, as I mentioned earlier, they act differently. They're like coincidence detectors. They're recognizing a state of the region. Typically, we went again back to the neuroscience and said, well, how many synapses should they have? And it turns out on a neuron, somewhere between 15 or 20 synapses is sufficient to get a dendritic segment to become active. So we started with that, and we said, that's a lot less than the kind of cells. I may have hundreds of cells or thousands of cells that I want to be able to recognize the state of. So I'm trying to predict my activity from an activity of 1,000 cells. And I'm only going to make connections to 15 of them. And you might, well, that could lead to an error, but it doesn't. Statistically, it's almost impossible, and this is the result of having very large distributed patterns. If you can recognize 10 active cells out of a very large sparse distributed pattern, that's almost guaranteed to give you the right result, meaning I'm recognizing the entire state. And so you only have to subsample here to get this to work. Again, as I mentioned, we form synapses from a potential synapse pool. So again, you can't connect to everything. You can only connect to a certain percentage of the cells, and that can change over time. Now, another thing we ran into, we wondered, could we, a typical neuron only has a dozen or 2 dozen of these dendritic regions. And we wanted to learn more patterns than that. And so we said, what happens if we overlay patterns on the same dendritic region? That is, we learn multiple patterns of 20 synapses at a time on the same dendritic region. And there's reason to believe that biology is doing this as well. And it turns out that you can say, well, I might have some errors. This dendrite is trying to look for a specific pattern of 15 cells, for example, 15 cells active. And now, but I might mix and match. I might take 5 from this pattern and 5 from that pattern. And what happens is you can overlay them. You can overlay them up to, we've been up to do it up to 20 times. And they don't get lost. They don't get confused. It's, again, a part of the sparse distributed representations. So it turns out a single cell can participate in many, many different sequences and representations, and yet still have the same feed-forward representation every time. These are the kind of things you see when you actually record from real cells in neocortex. You find out that the cells in a column tend to represent the same feed-forward input, but they seem to be very different in context, and they kind of have very different representations. And it's exactly what we expect to see here. Though our learning rules are pretty straightforward, the first thing we do is we learn on a segment-by-segment basis. And so it's not like it's just a synapse by synapse, it's a segment-by-segment basis. And we have to figure out which segments we're training. And basically, if a segment is active, meaning a distal dendrite was above threshold or the cell got enough feed-forward input, we said those are good synapses. We just want to reinforce them. We basically increase their permanence. We don't change their strength. We just increase the permanence. And we decrement the permanence of the guys who are not contributing. If a synapse is not contributing to this dendrite becoming active, it didn't seem to be very useful. So we modified them on a segment-by-segment basis. We also have to sort of make sure we connect to things that just were happening in previous time. We don't want to connect to just the current input. We want to connect to things that were just active momentarily before. And so we have to make sure we basically are training on cells that are in T-1. In a real biology, cells stay on for a long period of time. It's not an issue. In a computer model, we have to be a little bit more careful about it. And then when we modify the permanence, as I said, we increase the permanence for active synapses. We decrease the permanence for inactive synapses. We give a range of permanence like 0 to 1. And if you're above 0.2, you're a valid synapse. And if you're below 0.2, you're not. And that pretty much sums it up. And those are the rules you need to do. So let me try to give you a summary of all this. We needed to do the following things, and we did. We needed to achieve a variable order sequence memory. And if it's not clear to you, when we say variable order, you can use as much context as necessary. If the statistics say, I need to look back 1,000 notes in the song to make the next prediction, it'll do that. It's ideal for time-based and static inference. We've tried this on lots of different types of problems, recognizing both sequences and doing spatial inference in vision. It has a massive predictive ability. You can overlay multiple predictions at the same time. The system doesn't get confused on it. And it uses sparse distributed representations to make all this work. We've shown that it's high capacity. We have data on that. We've shown that it's high noise immunity. When I say noise immunity, I can do the following things. I can drop out a percentage of the cells and nothing gets confused. I can drop out synapses, nothing gets confused, up to a certain percentage. I can occlude inputs. I can add noise to the inputs. Of course, it depends on exactly what your data is and how much, but the typical kind of things we're looking at, it's very robust to noise. We've got it working in an online environment, so we can be doing inference and gradually shift to new representations if the statistics of the world change. It has a very deep biological mapping, and we have these self-adjusting representations. Now, typically in an academic talk, I would now show you all these charts and graphs to prove all this stuff. I can do that, But I'm not going to do it in the talk. First of all, because it's a lot of it, and it's very difficult to present. The talk, you have to explain what the data sets are, and you've got to explain what the exact numbers mean, and so on. So after in the Q&A, if you have a question on any particular part of this, how did we test capacity or something like that, ask me, and I'll try to address that. But anyway, so I'm not going to go into that at the moment. I'm just going to claim that we've modeled this stuff. We've done lots of different data sets. And some of you will be able to look at this mechanism and say, oh, I get it. I see why it's going to work. Some of you will be scratching your head going, huh? So what's next? So what's next for Numenta? There's two things for us. One is we're a commercial enterprise. And by the way, we're a commercial enterprise not because I want to be in a commercial enterprise. It's because I think it's actually a good way to get a lot of people working on interesting scientific problems. If you can show that you can make money at something, it's very fast. People react very quickly. Academia works slower. It works on its own little time frame. There's nothing wrong with that. But I think this is a great way of getting people to work on these problems. You put it in a commercial setting. We are going to apply these problems to data sets which are not classic AI problems, but problems where people have huge amounts of data with time-based statistics in it, and they don't know how to analyze that data. So here's some examples. We're working with a Spanish bank and a company in Mexico looking at credit card fraud. They have millions of transactions over time, and we're trying to figure out how do you discover the temporal and spatial statistics in those patterns to determine anomalies and fraud detection. There's lots of questions that come in your head, but one of the things you might say is, well, how do I do that? Well, you have to take a credit card record, which we have millions of them over time, and you have to turn it into a sparse or into a distributed representation that's amenable for this kind of algorithm, but it looks doable. We have a lot of companies that come to us with large sensory environments, like sensors on buildings or in factories, and they're swimming in data, and they don't really know how to model that data. And they want to say, hey, can you tell me how to model it, how to do prediction and inference on it? We've done some things with web prediction, just like watching someone move through a website. Can I predict where they're going to go next? And you can, with statistically, But we're interested in the research as well. We love this stuff. We love the neuroscience aspects of this. First of all, I want to point out that the full documentation for these algorithms plus pseudocode is posted on our website. We put it there Wednesday. And so if there's any of you in the audience who be like, hey, I want to play around with this, you can do it. It's not that hard to implement this stuff. We're going to update that documentation as we make changes to it. We're going to release some software for this in 2011. I don't have a date. I don't know exactly what it's going to look like yet. But we're going to make that available. All of this is free for anything we do for free for research purposes for all time. If you wanted to do something that's commercial based, then we might ask you to take a license. But at the moment, we're not even close to that. We're just saying, let's go for it. Let's experiment with this stuff. Here's my one little advertisement. We love interns. We've had a couple of interns from UIUC. Lydia Majur is someplace in the audience here. Where's she? Raise your hand, Lydia. Oh, she's hiding out. There she is. Edgar, I don't know if Edgar's here. We had another guy, Edgar. Anyway, we love interns. If you want to come out to California and spend a summer with us, let me know. We also have a couple full-time positions available. That's my email address for any questions you might have about anything. I talked. Second part of my title of my talk was, the impact on future machine intelligence. And so I'm just going to wax a little philosophically here for a moment. I think understanding how the brain works and building machines that work on it is the greatest thing anyone can be doing in the scientific world today. And it's got so many. It's like got every element you could possibly want. It's understanding who we are, humans. It is figuring out why we do what we do. It can lead to great social benefits for helping us out both as humans in medicine and understanding disease and education, and as well as making amazing technology. And if I think about the amazing technology, go back to the beginning of the computer era. You weren't there, but you go back to 1940s and you read these guys. You read about Turing and you read about von Neumann and the people who started the computing revolution. They knew they were onto something big. They didn't know where it was going to go. They had no idea that we're going to end up with cell phones and and GPSs and things like that. But they knew that they were onto a really basic idea. And we've had 60, 70 years of just tremendous innovation and benefit for society coming out of that. We are at that point right now. It's not going to happen 20 years from now. It's right now. And so the reason I come and give talks like this is I'm trying to catch 2 or 3 of you in the audience right now who have not decided what you're going to do in your career. I'm telling you, do this, okay? That's the end of my formal comments. Thank you very much. So, did you want to?
I think we'll let Jeff field questions. It makes it much more efficient.
Yeah.
And I will interrupt if we start to run too
Oh,
long.
okay. You'll just make sure we don't control it. And if you do need to leave, go ahead and leave. I don't care. I'd be happy to take questions about anything, and I'll try to keep us on track. And so who wants to be brave and be the first one to raise their hand? All right, down here. Yes, Neil.
You did. That was wonderful. Thank you very much. Slightly ill-formed question. You distinguish between the current input and context, and you talk about how sparse the response to the actual input is as compared, for example, to the portion of it that's predicting the next piece. I'm wondering whether that's the same or different from talking about multiple scales in time of the representation.
Well,
So if you think about the representation, not just in this tiny time slice, but a longer time slice, you're getting the input is extended over time. It includes both the immediate response to it and the prediction as it changes.
So Neil, so I want to repeat your question, but I didn't catch the whole thing. So just try one more time. Sorry.
I'm wondering whether you're thinking of your representation, your sparse representations, over a very fixed and small time scale, or whether you can entertain thinking of them at multiple time scales, which would encompass both the input and what you're calling the input and what you're calling the context.
So the question is related to, and I still don't understand the question, so I'm going to try to answer it, and then we're going to relate to it. The question is about, is this prediction over multiple, the representations, especially representations, over multiple time scales, or?
I'll give one more shot.
Okay, I'm sorry. I'm usually really good at this, and you and I were clicking yesterday, so I must have eaten something bad this morning.
You could imagine the input at this moment in time you see an individual object and it's context free in the sense that you don't have any information about what preceded and what's to follow. And so you can distinguish between the input itself and how it's represented and the larger context. And you talk about that happening, in fact, in different layers.
No, okay, so here's what, let me try to clarify that. I think the question is really, I'm not sure if it's a question, but I'm going to try to answer a question that I can answer. You know, at that point in time, you have an input independent of context. So, you know, there's something coming in my ears, there's something coming in my eyes. My retina doesn't know what happened a minute ago. And I have to form a representation of that, right? That's the independent of context. And then I have to form I need now to form a representation of that in context, meaning it has to be different. I need to, at some point in time, I have to have a different representation, which is representing that input in the previous states. I'm looking for guidance from you, Neil.
And can't you just think of them as multiple lengths of time, multiple time frames over which you're representing?
And so the question is, can I think of them as multiple time frames over which the representation? So I'm still confused. Let me just clarify. There's no concept of, I'm not relying on some particular cell to stay active for some context. It's not what's required. What's required is that at any point in time, I have a unique representation. So let me tell you, let's say I'm learning a melody. I'm learning Beethoven's Fifth, okay? Bum, 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 right? There's repeated sequences in there, subsequences. That first phrase, bum, 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 is repeated exactly a few seconds later. That sequence is repeated a few seconds later. I do not get confused as to where I'm going next after the first one versus after the second one. That tells me, even though I have the exact same pattern coming in my ear, I must have a different representation when that pattern comes in later because I'm in the context of the song. We don't keep anything on. There's no cells that are on during that time. It's just that every point in time, I have a unique representation. So the fifth time that thing occurs in that sequence, I have the same set of columns. If it's a spatial pattern, I'd have the same set of columns active, but I have a different set of cells in those columns active saying, this is that note in the 32nd appearance. And it turns you're just doing this all the time. That's why it's so important to have a very large number of ways of representing the same input. You know, if I am able to memorize that melody, and I'm not without a hierarchy, and I can do this. We've done this. We can memorize very long sequences, and the only difference between sequence A and sequence B is something that occurred in the beginning. They'll still be completely clear at the end. And by the way, even if I have a prediction, I didn't mention this. If I have a prediction, so it's a unique set of cells in the columns, I can say, well, what note is that? I just drive down the column and I say, oh, I can tell you what note it is. Even though I have a unique representation for the 57th interval in this song, I can still tell you it's an interval of a fifth. And so I can go back and forth between these sort of representations of the feed forward, out of context, in context, out of context again as you go up and down. This was the key, actually. This whole mechanism was the key for me personally, breaking through some log jams that we had. And so, Neil, I hope I got to that. Okay. They won't all be that hard. Yeah. Yes. I'm sorry, I've forgotten your name. Yes.
Okay. In your model, you started from talking about the different brain regions and this sort of network of brain regions, and talking about their hierarchical roles and so on and so forth. However, in the second part, when you talked about your practical application, you talk only about one region only.
Yeah.
So, what is the relationship between those two?
Okay, so the question is, in the beginning I say how the different regions of the cortex do different things. And then in the end, I'm just talking about one region, right? So I tried to make this connection. Perhaps I didn't do a very good job at it. I tried to say, there was a slide, I said, look, all these problems, whether it's speech, whether it's vision or hearing or touch, first of all, we know they work on common algorithms, but the algorithms, this mechanism here underlies all those algorithms. I can apply this audition, I can apply it to hearing, I can apply it to touch. They're all the same problem. You have a set of sensory data coming in, you have often a topology on that, and you're trying to discover the spatial and temporal statistics in that, and it doesn't really matter what the problem is. So I can apply this to bank fraud data, I can apply it to vision, and I can apply it to hearing, and I can apply it to motor problems. That's the basic, that was essentially the argument that was made 30 years ago by Mountcastle. If you can illuminate the core fundamental, what is the same between all those different problems? Now, at different levels in the hierarchy are the following. So if I take a vision problem, for example, and the first layer in the vision area, I apply one of these layers to it, it's going to say, all right, the best I can do is represent small areas of, it'll do this. It'll say, I'll represent small areas of the visual space. I'll look for the common spatial patterns. Those will be like edge detectors and corners and things like we see. And then I'll learn for sequences of those. Those will be like complex cells. The next level of the hierarchy is looking at, I have convergence and it's looking for combinations of those, but it's the exact same algorithm. And as long as I can do the convergence and the temporal, I can form a slower representation of these guys as I go up the hierarchy, then I will have, I'll end up with slower representations of common patterns over the entire input space. And that's independent of modality. It just has nothing about the modality there. So it's a generic learning algorithm for figuring out, what's going on in a sensory space.
Now, related to that, is it possible that those regions may have a relatively similar architecture, but some small, not even small, some variations?
So the question is it possible, even though they have a similar architecture, that those regions actually have something unique about them that makes them different?
Like some parameter?
Yeah, some parameter, or some magic cell, or some extra little chemical, so here's what we know. Okay, this is the half full, half empty sort of argument in neuroscience, right? We know that there are variations of them. We know when you go to region, you'll find some regions have a denser representation, more cells in layer 3 and some less cells in here. I mentioned layer 4 goes away. We know that there's a striate layer 4 in V1. We know that there's no layer 4 in M1. There are variations of a sort. And the question is, do you think those variations are the key, or do you think they're just variations due to the statistics of the data that information is that region is processing? There's some evidence, for example, striate V1, of course, is going to be genetically determined. However, the vast majority of evidence outside of primary sensory cortex, especially outside of V1, is that those variations are learned. That is, the statistics themselves determine how much, how many cells you allocate to different types of parts of this and the densities and things like that. And we know that because we know for certain that almost all cortical regions can be repositioned to do something else. That started with Magarka-Sur, and now we know what happens in humans, and so there's nothing visual about visual cortex. That's what Vernon Mountcastle's exact words were. There's nothing visual about visual cortex, there's nothing motor about motor cortex. Some people don't believe that. It used to be a majority of people didn't believe it. Now the vast majority of people do believe it, and there's a minority who don't. But it's part of our work. Our work is assuming, let's start with the basic algorithm and then figure flavors of it and variations of it in different contexts. Yes?
I didn't understand a few issues, maybe you can clarify some of them. How do you read outputs from your system, how you interpret this output, and how you stimulate this.
Okay, the question is, he said, how do I feed inputs into this? How do I read outputs from this?
Well, inputs I understood, but outputs.
Oh, outputs.
Some output cells.
Yes, it turns out all the cells are output cells. This actually is true in the cortex too. If you look at like every layer 3 cell, it projects externally to that region. It leaves and it either projects to the next higher region of the cortex, it also bifurcates and goes elsewhere. But every one of those cells, the mass of them is the output of a region. So it's a very large distributed pattern. And you'll see here that it's no longer as sparse as when I turn it into a sparse representation. I then do the prediction, it becomes less sparse because all the yellow cells and the red cells are the output of this region. And then that becomes the input to the next region, and it gets sparsified again, if that's a word. So it is the vector of all those cells' activities at this point in time, which is the output region. That's both biological and that's in our model.
But how do you interpret this output?
How do you interpret the output?
To detect the fraud with credit cards.
Okay. So how do we detect that? But there's several answers to that. What do I do with this output, right? One simple answer is I can, first of all, detect anomalies. I don't have to do anything special. I can just see how much ill prediction I've got here. And I can see what that relates to. But typically what we will do, which is not what's happening in a brain, okay, what we will do is we'll put a classifier on top of this thing. If we're at the top of the hierarchy here, we can put a classifier in it, an SVM classifier, or nearest neighbor, whatever you want. We can put a classifier in it and we can read out the answer. We can say, okay, if we're trying to do an inference and categorize it, we'll just do that. That doesn't work if you're not at it doesn't work well if you're not at the top of the hierarchy. If you're trying to do that on top of V1, you're not going to get very good results. So we have a whole bunch of metrics we use to determine how well this is working. But in the end, if you want to read it out, which you can't do out of a human, you would go up to the top here and we put a classifier in it. In a human, what happens is, don't get into the hippocampus for a moment, but the human, what happens, I will feed that information. I can take this pattern and I can feed it back down a motor, and I can make speech, and I can then unfold. This becomes the representation of an unfolding speech pattern or some motor behavior pattern. But from our point of view, we'll just stick a classifier on the top.
And do you stimulate it somehow? Let's say if I touch something hot, I feel pain, so next time I don't do it. So there is some stimulus from outside world. Do you have some kind of stimulus?
You say, is there a stimulus from the outside world that tells me, like, if I touch something, it's hot, this is good, and it's not good? So what's the answer to that question? How do I want to answer that? Well, in some sense, we don't do it. There are two answers to that question. Sometimes people ask that question, they mean two different things. That's why I'm thinking about it. In one case, if I'm just trying to classify something and say, oh, this is a dog and that's a cat, I can just do that in the classifier level. I just put into the classifier saying, hey, this set of patterns are all dog patterns, this set of patterns are all cat patterns. There's a second way that people interpret this, which is to say, there's an emotional content. How do I know that some things are more important than others? And I'm not sure if that's what you're asking about. But we don't do that here. Emotion, essentially, we would do that by affecting the permanence of synapses under certain conditions. So I would say, okay, not only going to learn, on this particular pattern, I'm going to learn very rapidly, but I'm going to make these synapses very permanent, so you're not going to forget about them. We can do that, but we don't really model emotional salience at the moment. Right now, we're just modeling the statistical data on a sensory organ, trying to fill a model of typical, non-sentient, non-emotional content. Can I just learn the things? But in the end result, at the very top, I could just put the classifier in and label the classifier.
What happens if the system makes a wrong prediction? Do you tell the system?
If the system makes a wrong prediction, do I tell it? Well, it knows if what's wrong. Wrong could be just noise, right? I'm not going to make. So a wrong prediction isn't always wrong. It just says I have some novel data coming in which didn't match. What I do with that is sort of independent of the model I just said here, right? If I say, oh, it's wrong, it should have been right, I'll train the classifier. But what's wrong in one component in time is not wrong in another. So for example, let's say I learn a sequence, and the sequence is A, B, C, D, and then there's noise, right? So it learns to make no prediction about that. Right, so it says, like, I have no expectation, but now all of a sudden it starts saying A, B, C, D, and E start showing up more and more again. So now all of a sudden it says, I'm going to extend that to E. And then what if I saw F? Well, initially it'll say F is wrong, but then if you train enough, let's say maybe A, B, C, D, F is the right thing. So it's not a simple answer of just like, oh, what's right or what's wrong. It's more about modeling the temporal data. Boy, you guys have tough questions. I mean, they take a long time to answer. I'll take this. One more, and then we'll hang around a little bit. Yeah, you scored.
I was just going to ask what the difference was between the activity of a unit that's in this predictive state and a unit that's in this coding state, and how you prevent your algorithm that does this sparse pruning and makes this sparse representation from choosing one of those states, either the prediction or
All right, so this is a very interesting question, and I decided not to talk about it because I didn't think most people would understand it. So now's my invitation to give you one last interesting piece here. We have these two states of the cells, the predictive state. I'm not going to rephrase your question. I'll just talk about the issue of differentiating predictive state and active state and how do you know how to use them. Here's the issue. So we have a set of cells that become active from feed-forward input. We want to propagate we want to now make a bunch of other cells active in anticipation. However, what if I just made those other cells active? Well, wouldn't they then force new cells to become active and then new cells to become active? And you would have a runaway prediction. So you can't have that. So this is one of our constraints. And we said, how could we do this? So here's the hypothesis, and I'll walk you through it completely. The hypothesis is that when a cell's in a very rapid firing state, whether it's a burst or it's just a rapid firing state, we're not going to make that distinction. That's physiologically different than if it's in a slower state. Now let's think about that. It turns out that let's take the feed-forward input on the proximal dendrite. Okay. Now the feed-forward input on the proximal dendrite is the union of all these guys, the slower guys and the fast guys. Well, if you look at the literature on proximal dendrites, they're kind of insensitive to rapid rates on a particular synapse. In fact, it almost hurts. And so the first spike that comes in produces an internal potential change. And the second spike that comes in right after it is decremented quite a bit. So a bursting thing wouldn't have much of an effect. And so therefore, all these types of cells, whether they're slow or fast, actually have the same effect on the proximal dendrite. And what we want to do then is on the distal dendrite, we want to distinguish between them. We only want the fast one to matter and the slow one not to matter. Because the slow one is a prediction. We don't want to propagate predictions. And you see those same type of behaviors on the thin dendrite segments. That is, synapses that are active very rapidly actually do summate nonlinearly, but slower ones will have less of effect. So you actually are biased towards the things that are fastly spiking and unbiased toward the ones that don't. So the physiology matches that, and it's a prediction from this theory because it is a prediction that these are going to really make a difference here. And but we need it theoretically. But biology sort of matched it as well. So there's a little subtlety that goes on there. We have to sort of keep these things distinct, whether it's a bursting and non-bursting or just fast or slow. A little remains. But I've talked to some neuroscientists about this, about the bursting concepts, and who study it. Not many people do. But it looks like all pyramidal cells have this sort of burst mode that occurs in the beginning of innovation. And that actually fits what we want. We want it to burst when the feed-forward input comes in. So that was a deep question. I hope you got some of the answer. If not, I can try to explain it later. We're out of our time, so thank you for sticking around.
Yeah.
Appreciate it.